Okay, now let's take a look at antioxidant protection using the glutathione system. The reason this is important is because we are going to see a very important use for the NADPH that we have generated from the pentose phosphate pathway. So oxidative damage to cells occurs in many different ways, but a primary pathway we can look at is involving oxygen. So if oxygen accepts an electron in an uncontrolled oxidative state, oxidative reaction, we are going to generate a superoxide free radical. Now this extra electron is an unpaired electron, and so this can attack many different compounds, including lipids, proteins, carbohydrates, nucleic acids. The superoxide anion, can also be converted into hydrogen peroxide. Once we have hydrogen peroxide, we can quickly generate hydroxide free radicals. So we go from peroxide to hydroxide radical. Now, where do these come from? Any cell that has oxygen in it can undergo this reaction and generate these free radicals. So, for example, cells with mitochondria, with the electron transport chain, electron transport is typically a very well-controlled process. But anytime electrons become uncontrolled, we have the makings for a perfect storm because we have oxygen as the final electron acceptor. And if the oxygen is not converted to water by complex 4 of the electron transport chain, we could generate a superoxide radical. This also occurs in red blood cells. Now, red blood cells do not have an electron transport chain, but they do have a very high concentration of oxygen being carried bound to hemoglobin. So any kind of oxidative stress can trigger free radical formation. So one of the major ways of taking care of this radical, to eliminate this radical, is through what's called the glutathione antioxidant system. Glutathione is a tripeptide of glutamic acid, cysteine, and alanine. Glycine. The important feature of glutathione is that the cysteine residue has a free sulfur, a half group of free thiol. This will allow this molecule to undergo a redox reaction. So if we take glutathione with our peroxide, it is able to take the peroxide molecule and convert it to water. The enzyme that does this is called glutathione peroxidase. And in order to perform this reaction, to take care of that unpaired electron that forms in the hydroxide radical from the peroxide, we are going to oxidize glutathione. So glutathione, cysteine residue, and now we are going to use a second glutathione molecule that will be donating the electron to neutralize that free electron to form water. Now we only have a certain amount of glutathione in the cell, and so that means we need to regenerate the oxidized form of glutathione back into its reduced form. This is done by glutathione reductase. So again, this is another redox reaction. And in this case, we need to reduce the oxidized pair of glutathione into its reduced form. So that means we need to oxidize something else to provide the electron, and this is where NADPH comes in. So NADPH will be providing the electrons in order to reduce the glutathione by glutathione reductase to regenerate free glutathione that can now participate in additional reactions to neutralize peroxide and hydroxide radicals. The NADPH, of course, is generated from the pentose phosphate pathway. Now this leads us to a discussion on hemolytic anemia. Hemolytic anemia means lysis of red blood cells leading to anemia. So how do these red blood cells lyse? Well, individuals that suffer from this will have a glucose 6-phosphate dehydrogenase deficiency, which means that this dehydrogenase enzyme is either very low expression or absent. And recall that this is the entry step into the pentose phosphate pathway. So if we have a deficiency in this enzyme, we are unable to produce NADPH, and we will have an NADPH deficiency. With an NADPH deficiency, we will not be able to reduce the oxidized form of glutathione, so the glutathione that is present will able to undergo one round of neutralization of the peroxide into water, become oxidized and dimerized by the cysteine bridge, and then we're stuck. So these individuals will have, with this deficiency, will have a dramatic increase in the hydroxide radicals. Now, something that hydroxide radicals love to attack are lipids. Primarily think about the phospholipids of the cell membrane. Recall that this phospholipids have a glycerol backbone. We have a fatty acid chains coming off of the first two carbons, and then we have a phosphate group with a head group. Phospholipids can have fatty acids with variable double bonds, a variable number of double bonds. Double bonds are a highly reactive target for hydroxide free radicals because this unpaired electron will go after those double bonds. If this covalently binds to that double bond in the fatty acid, we are now introducing a polar group into the membrane of red blood cells. This will, of course, disrupt the hydrophobic bonding that holds the membrane together, leading to membrane rupture. Hence, lysis of red blood cells leading to anemia. The other problem is favism. Fava beans have a large amount, a large concentration of oxidative molecules. So what this means is that with increased oxidative stress, because of ingesting fava beans and their components that lead to this, we have an increased amount of hydroxide radicals. For individuals that can have 6-glucose-6-phosphate dehydrogenase deficiency, they are compromised in their antioxidant properties, and so these free radicals can dramatically increase their hemolytic anemia. The other issue with this oxidative stress, looking at the red blood cells, is going from oxyhemoglobin, which is oxygen-bound hemoglobin in the red blood cell, undergoing oxidative damage so that the oxygen is released from the hemoglobin as a superoxide anion. This will lead to an accumulation of methemoglobin, which is unoxygenated, and these free radicals can form, which can attack the methemoglobin. By this covalent linkage of these free radicals, we now disrupt the conformation of the protein, which is methemoglobin. Disruption of the conformation causes aggregation, and aggregation in red blood cells will lead to the Heinz bodies, which are insoluble aggregates of hemoglobin due to the oxidative stress and oxidative damage.